produce hydrogen production. I'm Gawel Soloski from Bengal University, and I will shortly describe my hydrogen production from product fermentation, from sour cabbage, uh, proteins, and potatoes. And these are matters. These are three matters which will be introduced to the topic. What is dark fermentation? Dark fermentation is a special case of anaerobic digestions. This is what is anaerobic digestive bacterial conversion of organic waste into methane, and dark fermentation is closing this process in hydrogen production. So because this stage before occurring methane production, because dark fermentation we have three stages or two stages if it's not complex, but usually our wastes are very complex material, for example polysaccharides and polyglycerol proteins are very multiple chains. So firstly we hydrolyze this bacteria, then if we get short organic material like glucose or fructose, it's enough to be digested by bacteria. So in this stage, because bacteria like such saccharides or saccharides, is the optimal products. However, proteins also, in this case, occurs also in case of proteins is also occurring, but mostly research is on polysaccharides, and this glucose is transferred by this bacteria into three possible pathways. These three possible pathways are acetic pathway, which is the most favorable because produce 4 moles of hydrogen per mole of glucose, but is from Gibbs energy the least probably, so not occurring very often. Other is propionic pathway, and the most often is butyric pathway. So for glucose is occurring producing one butyric acid molecule and 2 molecules of hydrogen. So this producing of butyric pathways is most common. And why this hydrogen is so important? Because we have our fuel are concentrated now. Our hydrogen production is concentrated now on production from fossil fuels, and fossil fuels are going to be depleted soon. In 2050, with 2030, we have no oil besides Saudi Arabia. And until 2090, we have lack of everywhere of oil, and the gas also will be depleted. So, and we have big trouble of our waste. So we have to deal two problems, and dark fermentation is one of the possible solution, because it takes waste that needs to be utilized and to and produces hydrogen that we have to replace in our conversion method. Is this process suitable to medicine? Yes, because it naturally occurs when some bacteria will be decomposing some bodies of fracture. And sickness, for example, gangrene, is the case that bacteria makes our in gangrene this bacteria hydrogen producing, because in hydrogen this bacteria feeds better, mostly often in mesophilic and conditions. Some are in extremophilic, but most common is mesophilic. And in case of gangrene, the human body with its 36 to 40 degrees Celsius is optimal placed for thermophilic bacteria. And in case of gangrene, it can be it occurs, and if is so much can be hydrogen production that in case of body of Henry VIII, the King of England in the 16th century, their body exploded. It is these bacteria are also causing the tooth collapse, because as I mentioned before, saccharides are very good for this source of eating source, substrate, feeding source. So if you don't clean your teeth properly, those bacteria will come to your teeth, because, however, the anaerobic, the small addition of oxygen is useful for them, and eating this rest of your deal of your meal for sugar, they will produce hydrogen, and this hydrogen will make your teeth collapse. So yes, it's something combined with medicine, so we can design the conditions that optimal for bacteria, so we can produce hydrogen, and these conditions, so we can avoid the bacteria curing in case of medicine. And this production of hydrogen is, in my case, I was checking 3 cells, right? Mainly some cabbage. That is a cabbage from cabbage initially digested by lactic acid bacteria. The sour cabbage is, in Poland is sour cabbage, in Korea is kimchi, is often dish in our countries where it's occurs, because it was good to store, because the sour cabbage was getting rotten. So it was good. And this, we checked two cases of sour cabbage, more digested and initially digested, and this less digested sour cabbage, in case of hydrogen production, was better than more digested sour cabbage. But in case of methane production, if we wanted to produce methane, not hydrogen, it was reverse situation. But coming back to duck fermentation, also we check the gelatin. And the case is in gelatin side that because duck ferment, we need to measure the gas, and was that in this gas from produced by this bacteria, which we catch by sewage because we kept our bacteria in special jars, glass jars, in water bath, which was keeping our temperature of 36 degrees Celsius, and these bacteria were coming by ducts and from cylinders. In these cylinders were filled also in water in other stuff, and this gas was pushed by this water, and this gas was measured by gas analyzer or gas chromatography. And gas chromatography, in case of every case of our duct fermentation, was measuring concentration of gas, and beside hydrogen was hydrogen sulfide. In case of protein, we checked, and then for some cabbage that because that this wasn't reported, however, there was big gases depletion because was some gas that wasn't measured because it wasn't hydrogen, and it showed that because the hydrogen sulfide, when we calculated, was too high, that was from products. This hydrogen sulfide was from bacteria, because this bacteria during fermentation is because these bacteria to stop methanogenesis are usually stressed, and this stress bacteria is going to this is Saures bacteria sponge, and by this hydrogen produced action also occurs and hydrogen sulfide due to eating by them bacteria rest, and this is why hydrogen sulfide, because it was occurred. We calculate how much hydrogen sulfide was as result, and I calculated that it was too much, that it comes from substrate. So the between hydrogen also occurs some hydrogen sulfide production and carbon dioxide, which is common. And what's more, duct fermentation has some advantages in other conversion methods that can every kind of organic waste, many kinds of organic waste. It's not much selectivity, but much variety of foods, and can produce for 24 hours. The disadvantages is that it doesn't produce so much hydrogen like pyrolysis, but pyrolysis requires much energy, much heating energy. Pyrolysis is a combustion of waste by using heat to 500 to 600, while in duct fermentation we just need 30 to 60 degrees, 30 for 40 degrees in case of mesophagy, in 60 in terms of it. But and only method that is comparable to duct fermentation is microbial electrolysis, which other method of hydrogen production, which says that will decompose, because there are some bacteria from Pseudomonas that produce some voltage. So this Pseudomonas production bacteria is producing this if you put some bacteria, they can decompose, for example, ammonia. And this is also only quite comparable method to duct fermentation, because otherwise biophotolysis or photofermentation, they need, beside substrate and bacteria, also light, so very hardly to control. Duct fermentation is quite simple, because you need to vary only bacteria and food, because these are two variables. So this is the advantage, that there are few variables, so easily control process. So this is all that I wanted to say. Thank you very much. for listening to me. Free to comments.